So today I am happy to introduce Donya Shodja. She is a PhD student at the University of Pittsburgh in the lab of Mark Rebeiz. She previously did her Master's of Science at Oakland University, and she studied under Dr. Shailesh Lal, looking at the pre-RNA splicing factor involved in found in maize and how it's involved in seed development. And she did her Bachelor's of Science in Plant Biology at the Shahid Beheshti University in Iran. And she has won a number of awards. She's probably one of those superstar grad students, which I can actually attest to because I overlapped with Donya a little bit when I was still a grad student. And she is a fellowship awardee of the Andrew Mellon Predoctoral Fellowship, and she also won the Ivy McManus Award at the University of Pittsburgh, which means you're one of the top grad students in your class. So that's exciting. And I'm really excited to hear what Donya is going to talk about and what she's been up to in the Rebeiz lab. So without further ado, Donya, do you want to go ahead and start your seminar?
Right. Thank you so much, Christine, for your introduction, and also all the organizers for having me. I'm very excited to talk about my project with you today. So I'll go ahead and start with a very broad question that I'm interested in, and that is the origination of morphological novelties. And so what I mean by morphological novelties are anatomical structures that are not homologous to any structure in their ancestral species. So these are like the tusk of the narwhal, feathers in birds, or the turtle's shell. At one point during evolution, these structures were all new. But what is the genetic basis of forming a new structure? So we know that despite this vast diversity of shapes and forms, that the major signaling pathways, which are shared and conserved, provide instruction for their formation. So that raises the question of how are these signaling pathways deployed to new contexts to form such novelties. So this is really a problem of regulation. We know that there's a huge role for gene regulation to control when and where the relevant genes are expressed. And so understanding changes in development to make something new requires an understanding of changes in regulation. And so just to elaborate a little bit here, we're talking about forming new structures, so say the shell of a turtle, and that requires cells to differentiate during embryonic development. And a group of molecules that are necessary for this process, allowing cells to communicate and differentiate, are components of developmental signaling pathways. So we know that signaling pathways are important for forming novelties when we look at a turtle embryo where the turtle shell is going to form, we can see FGF8 mRNA expression, which is a signaling pathway ligand that's in spatial association with the cells that will later form the shell, so in these purple dots here. So we know that signaling pathways are responsible for turning on the right genes in a very specific spatiotemporal manner. And despite having many instances of this association, we currently don't understand the architecture of the loci important for regulating these signaling pathways, and then how that regulation has evolved. So we really need a system where we can find and manipulate these signaling events and then be able to trace their evolutionary history. And so to do this, I use the fruit fly Drosophila as a model, and particularly a recently evolved structure on the male genitalia called the posterior lobe. So that's the structure outlined here in blue. It is a cuticular outgrowth that's known to be a clasping structure during copulation. And what's nice about this system is that it's recently evolved, and so species exist that have a similar looking genitalia but without a lobe. So here I'm pointing out two other structures that are conserved, namely the lateral plate here in dark blue and the clasper in yellow. So these exist in all male fruit flies. And shown here at the bottom are dissected cuticles of lobed and non-lobed species. So we see that the lobe is unique to the melanogaster clade, and then we have non-lobe species, for example, Ananaceae and Biarmapes, that lack a lobe. So we can use our model organism, Drosophila melanogaster, to study the development of the lobe and then make comparisons with non-lobe species. And just to quickly familiarize you, Drosophila has four distinct developmental stages in its life cycle. So that's the egg, the larva, the pupa, and then it undergoes metamorphosis to form the adult. And because these genital structures form during the pupal stage, a lot of the images I'll be showing you will be at various pupal stages. So here I'm showing you a pupal genitalia at a time point which most of the structures have differentiated. And green is showing the apical surfaces of the cells. And I want to point out those three structures that I showed on the previous slide and the adult. So here is the posterior lobe, the novel structure, which actually protrudes from the lateral plate in dark blue, which is adjacent to another clasping structure called the clasper in yellow. So the lateral plates and the claspers are ancestral structures, meaning that they're older and also found in non-lobe species. Okay, and so to understand how the posterior lobe evolved, the first step for me is to understand how it develops. And as I mentioned earlier, we know that the deployment of signaling pathways to new contexts have been implicated in forming novelties. And so, in search of key regulators of the posterior lobe, the lab had found that delta, a ligand of the Notch signaling pathway, is expressed near the developing lobe. So here is an immunofluorescent image of half a pupal genitalia for simplicity, where delta antibody is marking this broad expression pattern of delta adjacent to a developing lobe. Now, if you compare this expression with non-lobe species like Biarmapes and Ananaceae, you can see that while delta is expressed at the base of these conserved structures, it's only in a small dot-like pattern. So the spatially expanded pattern of delta is unique to lobed species, or melanogaster here. And importantly, our RNAi data demonstrates that delta is required for lobe development. So this is a wild type, a lobe in the adult. And as you can see, when we knock down delta using a tissue-specific driver, the lobe is almost gone. And if we overactivate the Notch pathway, it increases the size of the lobe. So this suggests that delta's expression is necessary for lobe development and that Notch signaling modulates the development of the posterior lobe. Okay, so a quick summary here, and then I'll introduce a more specific question. So we have a recently evolved structure, the posterior lobe. We know that delta's expanded expression is essential for lobe development. In non-lobed species, there's a small dot pattern of delta at the base of these conserved structures, which exists in both lobed and non-lobed species. So it seems like this expanded pattern, which is necessary for lobe development, is an elaboration of a pre-existing signaling center. So this brings me to my main question. So how has this expansion of delta evolved? So how has the regulation of delta changed for its deployment to the lobe-forming region? So delta is expressed in many, many different tissues in the body and also in multiple genital tissues. And so because I want to find the changes leading to this expansion, I need to find the lobe-associated enhancer first to then ask how it changed to drive the spatially expanded pattern. And so using GFP reporters in transgenic flies, we found two enhancers of delta that drive an expanded expression recapitulating delta's lobe activity. So I'm calling them enhancer 1 here and enhancer 2. And so even though these enhancers seem to be redundant and both drive a delta-like pattern, I noticed that they're different in their spatial activity. So here's an immunofluorescent image of half a pupal genitalia showing the endogenous pattern of delta. And interestingly, while the two enhancers overlap this pattern, their spatial activity seems slightly different from one another. So we see enhancer 1 recapitulates the entirety of this expanded pattern, whereas enhancer 2 only overlaps this top portion marked by the blue bracket. So just to quickly summarize, we have two enhancers of delta. They seem to be redundant, but are not completely overlapping in pattern. Nevertheless, they both drive an expanded pattern of delta. So we were curious to know how this expansion occurred. Was it due to changes within these enhancer elements themselves, or are the elements functionally conserved and that the major changes are upstream in the transcription factors regulating them? So basically what I'm asking is, have the enhancers themselves changed? And one way to answer this is to clone orthologous sequences of the enhancer from non-lobed species hooked up to GFP and insert the construct in melanogaster, a lobed fly. So if the enhancer drives GFP expression in an expanded pattern, it'll indicate that the non-lobed enhancer is actually responding to the transcription factors in melanogaster and that the enhancer itself is functional or pre-existing. But if the non-lobed sequence does not drive expanded expression, it'll suggest that the enhancer itself has probably changed, or that's a new enhancer. So what do the results look like? Just to remind you, this is the pattern of the melanogaster enhancer 1. And when we look at ananase sequence in a melanogaster background, it also drives an expanded pattern here, which indicates that this element is responding to the trans landscape of melanogaster, suggesting that the enhancer is functionally conserved. But what about enhancer 2? So to remind you, this is what the melanogaster sequence looks like, and if we insert the ananase sequence in melanogaster, it's not active, which suggests that this enhancer 2 is probably a new enhancer and that changes within the element itself have led to its broader activity. So we found that changes both within the delta locus as well as the transregulatory landscape have contributed to this expansion of delta. Okay, so to build on what we know so far, enhancer 1 is functionally conserved. We can call it pre-existing, and enhancer 2 is novel. Now, specifically talking about melanogaster, we know that they have differences in their spatial activity, but these redundant enhancers could also be different in timing. So remember, these images that I showed you so far were at pupal stages when most of the genital structures were formed, so later in development. And so I was curious to know when each enhancer first turns on to see if there's a difference in timing. And in the interest of time, I'm going to use our cartoon model instead of the data and say that not only do we see a spatial difference, but also a temporal one. So enhancer 2 is on much earlier in the pupa than enhancer 1. And so that made me wonder, when does enhancer 2 first turn on? How early? And so I kept going back in developmental time, and enhancer 2 was always on. So I just went ahead and looked at the earliest time I could, and that's in the third instar larva, so a completely different stage in the life cycle when the future structures are still at the imaginal disc stage. And this is about 3 days before pupal formation, so that's a long time in the fly life cycle. And as you might have already guessed, enhancer 2, but not enhancer 1, was active in the larval disc. And this to me was really interesting because this is way before even the precursors of the posterior lobe start to develop. So it's about 4 to 5 days before the lobe initiates its development. And that's a long time for the signaling center to be on, especially if it has a later role in forming the lobe. So I did in situs to look at delta mRNA in the disc of melanogaster, the lobed fly, as well as 2 non-lobed species. And what I saw was that this is, in fact, a conserved pattern, which was consistent with my prediction because this is so early on. To me, it suggests that this could serve an early conserved function that potentially has to do with forming an older structure. Okay, so we know that there's a conserved pattern in the larval disc. It's very early on, and we don't know what it's doing there. And that this expression persists even in non-lobed species at a later pupal stage. And so we found this very intriguing and wanted to know what this early activity is and why it persists. And one really great thing about these GFP reporters is that we can track the activity of the enhancer through early development. And an interesting fact about the fruit fly genitalia is that early on, the genital disc is actually inside out, and then it everts during development. And what was fascinating to me was that the cells we see here or actually track with the cells that undergo this eversion process and are the same cells that we see later in the late pupal stages. So it seems like these cells that undergo this essential process for genital formation are specified very early on in development. And so we wanted to test the function of this enhancer by taking it away and then looking at the morphology. And so to do this, we deleted each of the enhancers separately. But surprisingly, we did not see an effect on the posterior lobe. So this was a bit puzzling to us, and it made us wonder if we were perhaps missing anything. And so we went back to our delta locus to see if there were regions missing in our reporter collection. Furthermore, when we initially started screening the delta locus for enhancers, we had no idea that delta was on in the larval disc so early, and we had only been looking at later pupal time points to screen. But with the knowledge that we have now, it's completely possible that we've missed early enhancers in our screen. And so, long story short, we found another region downstream, a region upstream of delta, and 3 intronic regions that seem to drive larval disc activity in these lateral folds where the cells are marked. So there's a lot of redundancy in the locus. That's about 7 early enhancers, which, while very interesting, complicates our ability to perturb delta in a specific spatiotemporal manner. And so to summarize and conclude, we found that delta notch signaling plays a critical role in forming the posterior lobe, and the expression of delta in the lobe-forming region was actually built upon and added to a pre-existing expression pattern by changes both in cis and trans. So this project shows how signaling pathways involved in forming new structures may actually be elaborations of ancient signaling centers that exist before novelties appear during evolution. And so, in our conventional view of how novelties arise, perhaps we would have thought that a new enhancer evolves to drive a new expression pattern that then generates this novelty. But in reality, what we see is that the signaling center is built upon an older activity that's on for an extended period of time. Furthermore, we found that this pre-existing activity is regulated by a fairly complex apparatus of enhancers that then become associated with this new function. And so it suggests that when we're thinking about how novelties form, it might be a common phenomenon that new activities are added to already robust expression patterns. And lastly, I really want to emphasize the importance of time here. We wouldn't have captured all this redundancy if we hadn't gone back in time to the larval stage. So we found 7 enhancers of delta in the larva, and we have to investigate whether they remain active in the early or late pupa. And what we find remarkable about these redundant enhancers is that some genomic regions are patterning the cells over a prolonged period of time, while others are active only early or only late. And so this really highlights the importance of time when we're looking at such chromatin signatures. And with that, I thank you all for your attention and again for having me. I want to thank my really great mentor and advisor, Mark Rebeiz, and the entire Rebeiz lab. Also past members of the lab for their contribution to this project. Also many members of the Pitt Biology Department, which is a wonderful community, and especially my committee members for their great mentorship. And with that, I'd be happy to take questions if there's time.
Oh, sorry, I was muted. Great job. Thank you so much for a beautiful talk. We already have some questions coming in. I wanted to ask, so Tim Lee Watkins, I noticed your hand was up at the very beginning of the seminar. Do you still have a question? Oh, okay. They turned off their raised hand. So first I will ask a question from the Q&A box. And for those of you who haven't asked your question yet, feel free to type in the Q&A box or raise your hand. So the first question we have is from Natalia Kochanova, and she says, great talk. Did you by chance look at the signs of notch activity example like cleaved or nuclear notch in the area of delta expression?
Yes, great question. So we did look at the activity of notch by, I'm trying to see if I have my slide up here that I could pull up. Okay, so we use the expression of M beta, which is a canonical notch target and is highly conserved as a readout of notch activity. And so here in this image on the left, we see that notch is active. So where there's green, that indicates that notch is active. We see that there's notch activity flanking delta's expression. And we see that here in the posterior, developing posterior lobe, which is corroborated by our in situs looking at the mRNA activity of notch as well. And also, when we look at the activity of notch in non-lobe species, we see this tight kind of circle around where delta would be expressed. Because, like I mentioned, delta is expressed in this small dot-like pattern in non-lobed species. So we see that notch is actually active and it's doing something there ancestrally or in these non-lobed species.
Cool. We have another talk from Felix Jonas. He says, great talk. Are the changes in the genitalia important for mating and thus crucial for speciation?
A very interesting question. So I can answer for melanogaster. So in melanogaster, a one group has used laser ablation to basically perturb the perturb the lobe, and
Mhm.
those flies don't have successful mating in melanogaster. Obviously, in other species that lack a lobe, they have other mechanisms for mating.
Cool. Well, thank you so much.